Good morning, good afternoon, and good evening, everyone, and welcome to the Scientist.com and Cell Signaling Technology webinar titled Unlocking Spatial Insights, Accelerate Discoveries 70% Faster with SignalStar Multiplex IHC. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be the host today. We're very fortunate to be joined by Dr. Sarah Klein, Associate Director of the Multiplex Assays Group at CST, who will present their brand new SignalStar Multiplex IHC technology and show how it can be used for spatial biology research. And with that, I'd like to welcome Dr. Sarah Klein. Sarah, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thank you so much for having me. Hi, everybody. My name is Sarah Klein, and I'm the Associate Director for the Multiplex Assays Development Team at Cell Signaling Technology. Today, I'm very excited to present to you a novel technology that CST has created called SignalStar. It will help unlock spatial insights and accelerate discoveries up to 70% faster. So many of you are well-versed in spatial biology technologies. However, for those of you who are new to the field, you can think of spatial biology as creating a map. In a map of our towns, our cities, different lights indicate your house, your friends next door, or your family down the street. They indicate businesses that provide services to you, like grocery stores or coffee shops. Some neighborhoods are more rural and quiet, while others are bustling with people, traffic, and commerce. All of it an ecosystem that coexists and interacts and depends on one another. If the coffee shop you go to every day closes, you will change direction and go to a different one. This could make you late for work or ruin your day. Interaction, impact. In order to really understand you, your neighbor, your shop owner, you can't just look at each individually. Each of you are impacted by the world around you and by the people you interact with, and each of you in turn impacts the world, as too do the molecules and cells of the human body. Spatial biology is not just creating pretty pictures. Spatial biology is mapping cells, locating their neighbors, characterizing cellular neighborhoods, identifying cellular phenotype and function within their original context in order to better understand who these cells are, what they are doing, and how they are impacting their world around them. Their world, in this case, being human tissues. There are 37 trillion cells in the human body in 22 unique tissues. There are only 7.9 billion humans in the world. So imagine a map with thousands of times more people to consider than on Earth. There are more cells in the human body than there are stars in the Milky Way. So yes, human biology and disease is complex, and creating a map of these systems sounds formidable. But spatial biology technologies enable scientists to decipher this complex biology, create these maps, in order to understand molecules and cells and how they impact healthy tissue and disease biology. In disease biology. In the brain and tumor microenvironment figures you see here, you can see the roads or vessels, houses or tissues, and people or cells that make up the maps of these tissues. Each cell has a purpose and it has individuality just as humans do. We call this their phenotype. Each cell also has an impact on the cells around it, affecting the function and phenotype of those cells. Changes in the phenotype and function of one cell can change the cells surrounding them, and so on and so forth, leading to changes in the whole tissue, in the whole body, in their whole world. This is what happens during diseases such as Alzheimer's or cancers, and it can be mapped and therefore understood using spatial biology technologies. Here's an example by Ken Young's lab at MD Anderson, where they published an article in 2019 using multiplex immunohistochemistry, a spatial biology tool. They studied a large cohort of diffuse large B-cell lymphoma patients and found the presence and proximity of PD-1 and PD-L1 expressing cells was significantly associated with patient survival. Patients without PD-L1 or PD-1 interactions had a better prognosis than those with PD-1, PD-L1 interactions. Another study performed by Janice Taub's lab at Johns Hopkins utilized a 6-plex fluorescent immunohistochemistry assay to stratify patients who are more likely to respond to PD-1 blockade. Patients expressing more of these of the 6 markers simultaneously in tissue correlated with a good prognosis versus patients with only 1 or 2 of these markers correlated with a poor prognosis. These studies demonstrate the need for both spatial context and detection of multiple biomarkers simultaneously in tissue. There are many ways that you can perform multiplex immunohistochemistry in tissue, and many different different platforms and instruments and assays available out there currently. Some of these technologies utilize direct immunofluorescence. Direct immunofluorescence involves the use of a single antibody that is bound to its epitope and is directly conjugated to its reporter. Here showing a fluorescent reporter, but other technologies use metal ions, etc. Another way to detect multiple antigens in tissue is to use indirect immunofluorescence. This involves a primary antibody bound to its epitope, and then secondary antibodies that are conjugated to their reporter, or here showing fluorescence, that bind to the primary antibody. Next is deposition assays. This involves a primary antibody binding to its epitope, followed by secondary antibodies that have been conjugated to the horseradish peroxidase enzyme. The HRP enzyme then catalyzes a reaction either with a tyramide fluorophore or a diaminobenzidine for a chromogenic assay, and deposits the signal surrounding the epitope and antigen around in the same vicinity. And lastly, there's oligo-based technologies that utilize directly conjugated antibodies, again, this time to an oligonucleotide sequence, and specific complementary oligonucleotides act as the fluorescent or as the reporters for detecting the antibody. This can happen in a non-amplified fashion or in an amplified fashion. But there are limitations to all of these technologies. For example, for direct immunofluorescence, there is no amplification of the signal. There's only a small amount of signal that is produced with a direct antibody, a directly conjugated antibody. And this sometimes makes it impossible to detect targets that are very low expression within your tissue. Using indirect immunofluorescence and secondaries, this can amplify the signal a little bit. However, it's not always enough to be able to detect your target in tissue if it's, again, in low abundance. In addition, it has limitations in relation to the host species of the primary antibody, making it limited in the number of the plex size that you are able to create. For deposition assays, you are able, these are limited by the fact that it is a deposition assay and the signal is not bound to the antibody, but instead deposited around the antibody, which can sometimes result in epitope masking and a crowding effect, again, limiting the plex size that you're able to create. Again, there are many solutions out there right now, but many of what we've heard is there's still many of them that lack the flexibility to design your panel as you want or redesign your panel when needed for your experiment. They often require up to 6 months of optimization for your panels in order to create a multiplex assay. Because many of them are incompatible with the instrumentation that is currently in the laboratory. And because of the optimization, the lack of flexibility in the instrument compatibility, it can take a lot of time and resources in order to create a multiplex immunofluorescence or immunohistochemistry assay. In response to this, cell signaling technology created SignalStar that has the ability to alleviate many of these limitations and concerns and can act as your solution. So what is SignalStar Multiplex IHC? SignalStar is an up to 8-plex fluorescent IHC assay. It enables you to be able to detect 3 to 8 targets plus DAPI. In addition, you can choose these 3 to 8 targets, any of the 3 to 8 targets from a growing menu of IHCP validated antibodies from our CST catalog. So the antibodies that are used in SignalStar have undergone rigorous validation, both in chromogenic IHCP, the gold standard, as well as the SignalStar assay. It is, again, highly flexible without time-consuming panel optimization. So you can choose any 3 to 8 targets, put them in any configuration you want in your panel, and it works out of the box. You do not have to perform optimization of your panel in order to run your experiment. You can get straight to your results. And that is because CST has performed all of the validation in advance. I'm going to show you in this presentation all of the validation that was done in order to enable this type of flexibility, as well as demonstrate reproducibility across tissue on microarrays and various FFPE tissues. This assay offers signal amplification for low abundance targets, as we'll describe in the validation as well. We have protocols for the Bond RX so that you can automate your protocol and perform it hands-off, 6 hours runtime, hands-off on the Bond RX. You can also perform it manually at the bench as needed. The assay touts having cycling without concern for epitope loss, as all of the antibodies are applied at once. This is very important, as all of the antibodies are applied at once. You also can eliminate problems of epitope masking as well as crowding, as they have equal opportunity to bind to their epitopes. Each kit provides enough reagents for up to 10 slides. And there is a panel builder that enables seamless and easy panel design to be able to combine these targets in whatever fashion you like. Lastly, we are imaging, instrument, and analysis software compatible. Therefore, as long as the fluorescent imager has the capability to image the 488, 594, 647, and 750 channels, the imager will be compatible with this technology. So how does SignalStar work? SignalStar is, again, you add all of your antibodies at once. So you have up to 8 antibodies that you would add in your first primary incubation step, and you would amplify the signal and perform your first round of imaging. You would amplify the signal of the first 4 antibodies and then perform your first round of imaging. You would then remove the amplification without disturbing the tissue or the antibodies that have been bound from the very first step, and then amplify the second set of 4 antibodies. Therefore, you're actually imaging twice and aligning these 2 images to create the 8-plex experiment. I'll go into a little bit more detail about the protocol in the next slides. So as I mentioned, you would start with FFPE tissue that has been baked, dewaxed, rehydrated, and antigen retrieval has been, one set of antigen retrieval has been performed. You would then add all of your oligoconjugated antibodies that were conjugated by CST with a proprietary methodology, as we'll describe later. All of these antibodies are added at once, and then we follow with a mild fixation using 10% neutral buffered formalin to be able to confirm that the antibodies will stay on the tissue and alleviate your concerns of antibody shedding over the course of the assay. We then use complementary oligos that will hybridize to the conjugated tags on the first 4 antibodies and amplify the signal of those 4 antibodies. This is a controlled amplification. It's not a PCR-based amplification. It's all done at room temperature, and it is a very specific, controlled amount of amplification to confirm that we are not saturating our fluorescent signals in the 488, 594, 647, or 750 channels, but instead staying within that linear range of detection, and we'll show a little bit later about the dynamic range of the assay. Once the first 4 antibodies are amplified, we perform a ligation step that enables stability of the oligonucleotides, the fluorescent oligonucleotides. That enables scientists that you don't have to run to the instrument and image right away. You have up to 8 hours in order to image the technology because the fluorescence is maintained over time. And so you would achieve your first image in your first imaging round. And in the next round, you would remove your fluorescent amplification methodology, as you can see here in this image, confirming that the fluorescence is in fact removed. We use specific double-stranded DNase that is specific for double-stranded DNA oligonucleotides. So it does not impact your antibodies. It does not impact your tissue. It specifically cleaves the double-stranded oligo networks and removes them from the tissue. And then you would use complementary oligos and amplifying reporters in order to amplify the second signal as I described. And as you can see here, the oligos are again ligated for stability to enable more time for imaging. So in the end, you end up with 2 images that you then co-register using either open source software such as QUPath or purchasable software such as Halo to create the final 8-plex image that you see here. Here's an example of a non-small cell lung carcinoma that was imaged with a SignalStar 8-plex assay. And we broke out the images here on the right into individual images so that you can see, again, correct subcellular localization and target localization for this panel and for these targets and antibodies. In order for cell signaling technology to provide a multiplex immunohistochemistry assay that does not require optimization or validation in your hands, the reason we can do that is because we've done all of that up front, and it all starts with a good, highly validated IHCP antibody using the hallmarks of antibody validation. So each antibody is first validated in using a binary strategy where cells that express the target or cells that do not express the target are used to help validate target expression. Next, we moved on to a range strategy where we use tissues that are high, medium, or low expressing for the target in order to confirm in an orthogonal approach such as RNA, mRNA detection, that we're seeing the same thing in our chromogenic images as compared to the RNA-seq that has been performed by using like the Human Protein Atlas. We use heterologous strategies such as overexpression models to make sure that we're not getting cross-reactivity of an antibody across family members. We use multiple antibody strategy in the fact that when we start our development of our antibody, we start at about 960 clones that is then narrowed down over the course of our validation to 1 antibody. And so as part of that, we often end up with a couple antibodies that detect the protein and detect unique epitopes within the same protein. And therefore, we can confirm with one antibody that is detecting a unique epitope from the other antibody. If they have identical staining, then it helps us confirm, again, specificity of the antibodies. We use, again, orthogonal approaches such as mRNA and mass spec in order to confirm antibody staining, as well as complementary strategies such as peptide blocking of the epitope for the antibody's epitope, or phosphatase treatment to block a phosphoantibody binding. Once we have performed all of the hallmarks of validation, we release our IHCP validated antibody. We then would take that antibody and conjugate it using the methods I'll be showing you in the next few slides. We perform validations such as this both before the conjugation and after the conjugation. The conjugation itself is site-directed. It results in a higher average degree of labeling, which results in a higher fluorescent signal. And we perform lot-to-lot reproducibility studies to confirm lot-to-lot we're seeing the identical staining. So SignalStar conjugation results in a higher average degree of labeling. This is a proprietary chemistry. You can see here in this chromatogram where we've pushed the conjugation efficiency to result in a higher degree of labeling, more oligonucleotides per antibody. So you can see there's a small peak of unconjugated antibody. There's a peak here for only 1 oligonucleotide per antibody, 2, 3, and 4. Alternative approaches for conjugation result in much lower efficiency of conjugation, and again, with much more unconjugated antibody. So for our assay, we found that a higher degree of labeling results in more fluorescent oligos and therefore more fluorescent signal. So here's a comparison between the SignalStar conjugation methodology and an alternative approach. And you can see using this SignalStar conjugation and purification methods to result in a higher degree of labeling also results in more fluorescent signal, even at lower concentrations of the same antibody compared to the alternative at higher concentrations. And we've quantified that here on the right, showing that consistently SignalStar conjugations outperform the alternative. After validating our oligoconjugate, we move now into the SignalStar assay, and we perform validation across a variety of tissues and TMAs, as well as across large tissue sections at up to 2 centimeters to be able to look for uniformity of the assay across the tissue. We validate every single one of our antibodies through every single place within the Plex panel. So you can see here, for example, SERP alpha was placed in the 488 channel. In a serial section, it was placed in the 594, in another serial section, the 647, and then in the 750. And you can see across these serial sections, regardless of the channel in which it was placed, you see consistent percent positive cells for percent positive SERP alpha cells. Most of our antibodies, the majority of our antibodies, can be paired with all of these fluorophores. In addition, we validated each of our antibodies across imaging rounds to confirm that regardless of the imaging round, the antibody performs as expected. And you can see again, looking in serial sections, that the percent positive CD206 remains consistent across these serial sections, regardless of imaging round. And the same thing with CD68, the percent positive remain consistent. So antibodies can be used and have been validated to be used in either imaging round. We also validated and compared the assay being run in the manual protocol compared to the BondRx protocol. Here you can see multiple targets and multiple unique channels. So again, confirming reproducibility across the different fluorophores and across the different protocols for each of these targets. Each of our antibodies are also validated in Plex. And so here's an example of a 4-plex that we've performed with pancreatin, PD-L1, CD20, and CD8. Here's the entire tissue, the colorectal adenocarcinoma, the 4-plex, and then we've broken out the single-plex images for each of these targets and compared them to the gold standard chromogenic DAB. And we've done this quantitatively, where we've looked at percent positive cells in a matching field of view across serial sections. And so you can see reproducibility across replicates and compared to the DAB as well. We've also tested the flexibility of our assay in creating multiple unique panels and looking for consistency of a target across those unique panels. So here you can see we have 3 unique panels where PD-L1 was placed in the 647 channel in each panel. However, you can see here CD3 was in 750 in panel 1. It was swapped out for CD8 in 750 in panel 2, and then Ki67 and 488 in panel 3. And so regardless of these unique panels, we did not change the protocol. There was no change in antibody concentration. There was no change in any of the reagents that were that were used for these 3 panels. And we saw consistent percent positive here in blue bars across these serial sections compared in the 3 panels compared to the single stain and the DAB, as well as the mean fluorescence intensity here in the orange line. Lastly, we also validated for each of our targets. Again, we performed all of this validation in plex. So here's an example of a 7-plex. When we're doing these validations, we're looking for tissue localization, subcellular localization, and target co-localization to guarantee accuracy. One of the fortunate reasons for performing multiplex is that you get to look at multiple targets on the same cell. That can not only be used to help you validate the assay, but also in order to confirm that the assay is not causing epitope masking or crowding of these epitopes. And so you can see here with the SignalStar assay, we can detect PD1, CD3, granzyme B, and CD8 all on the small T cells that are within the tumor microenvironment here in white. And so the assay has alleviated concerns of epitope masking as well as, again, crowding. SignalStar, as I mentioned earlier, SignalStar offers amplification for detection of low abundance targets. So here's some data that we've put together in regarding to testing CD8 in the SignalStar assay. We compared the SignalStar assay to tyramide signal amplification, indirect immunofluorescence, and direct immunofluorescence. And you can see SignalStar matches the mean fluorescence intensity for tyramide signal amplification, which is much brighter than indirect and direct immunofluorescence. Now, for a target such as CD8 that's in such high abundance, we're seeing the same % positive cells in each of these methodologies, meaning direct and indirect immunofluorescence may be sufficient for detecting this type of target. However, a target like PD1, where it may be in low abundance in many of the tissues that are available, SignalStar is much, as we mentioned, much brighter than indirect and direct immunofluorescence. As you can see here in looking at the mean fluorescence intensity in comparison. However, when you look at the % positive cells, SignalStar has a much higher % positive cells that matches the chromogenic dab, whereas indirect and direct immunofluorescence are not detecting the same number of cells, indicating the data. We're not using these methodologies. You're not able to detect all of the cells that are present in the tissue. And so, as I said, you can, in this image here, this is a close-up of a tonsil where you can see PD1 low-expressing cells that are amplified by the SignalStar technology. You can see them in your tissue and quantify them, as I showed in the previous slide. But you also have the ability to detect a medium-expressing PDL1 cell as well as a high-expressing PD1 cell, indicating a wide dynamic range for this assay. The fluorescence is not saturated as with other technologies. SignalStar is also non-destructive. So these are not serial sections. These are the same section where we performed the multiplex assay, and we were able to then afterwards perform H&E staining. So the tissue is intact, and you're able to perform H&E and look at the morphology of the cells and morphology of the tissue after performing your multiplex assay. So here's a growing menu of targets that are available for SignalStar. We have many immuno-oncology targets that are highly relevant for many targets that are highly relevant for the immuno-oncology field, and many more to come. If you want the list, there's a link in the handouts tab for you to be able to see the list of targets here as well to take home with you. In addition, SignalStar has a panel builder that enables seamless experimental design. It is an easy 4-step process that you can walk through within minutes to be able to take our list of SignalStar validated antibodies and quickly put them into an 8-plex panel or up to 8-plex panel. You can choose from a recommended panel that's created by the panel builder, or you can fully customize the panel design as you like. You can then download those results as this is your experimental design for your multiplex assay. I recommend trying the panel builder today. There's a link in the handouts tab in this forum. SignalStar is compatible with the BondRx. So before you get started, if you are using the BondRx, we always recommend that you confirm that you have the reagents you need from Leica Biosystems in order to run this assay. All of the information you would need is within the protocols of the assay. We also confirm you set up the SignalStar protocol on the BondRx software so that you're ready to get up and running as soon as you receive your multiplex kit. SignalStar is also, as I mentioned earlier, is compatible with most fluorescent imagers. So important, please confirm your compatibility of the fluorescent channels, laser lines, and or filter sets with your imaging instrument provider. So again, in the protocol, we provide this table in order to help you confirm that the instrument is compatible with the assay. And here are some examples down below. So in summary, there are many benefits to the SignalStar Multiplex IHC assay. You can generate images in just 2 days. As I mentioned earlier, it's 6 hours per imaging round. So 6 hours on day 1, 6 hours on day 2 in order to perform this assay. You can redesign the panels easily without any need for optimization, as each antibody has been validated in all channels throughout the 8-plex. This technology amplifies your signal so that you can detect low abundance targets such as PD-1, especially if, or for example, if PD-L1 is in low expression in your tissue. You can use your existing instrumentation. There's no need to purchase a large instrument. Whatever fluorescent imager you have available to you should be compatible, or you can confirm the compatibility with the SignalStar assay. Again, I hope I've shown you today that these are results that you can trust, as we've performed all of the validation in-house at Cell Signaling Technology, and all of the antibodies available for this assay have been developed and optimized in-house. We've eliminated the antibody cycling by adding all of your antibodies at once in the first incubation step, and you can utilize the panel builder in order to design experiments faster. Lastly, I'll leave you with, for more information, again, in that handout section of this presentation, you can find the SignalStar brochure, which has a description of the SignalStar technology and some of our validation data that I've described today. In addition, there's a SignalStar flyer, which will have that list of SignalStar antibodies so that you can start looking to see which panel you'd like to create. There's also a link to 2 scientific posters that were presented at the Spatial Biology European Conference in September that you feel free to download and look over more of our validation data and how we've optimized this assay. There's a link to the panel builder, as well as a link to more SignalStar resources to help you better understand this technology. And thank you for your time. I'd like to open it up to any questions.
Excellent. Well, thank you, Sarah, for a really fantastic presentation. Let's jump right in.
Okay, sounds good.
First one here, Sarah, what types of instruments are compatible with this assay, and how do I know if my imagers are compatible?
So yes, so as I mentioned in the talk, that this is a fluorescent assay that works with, utilizes the Alexa Fluor 488, the Alexa Fluor 594, Alexa Fluor 647, and 750. And so as long as your instrument is able to image those fluorescent channels, then it is compatible with this assay. So pretty much many of your fluorescent imagers have these channels available to them. In addition, in each of the protocols, we have a table that describes the specifications of these channels. So the emissions and the excitations for these fluorophores, as well as the compatible laser lines and the compatible filter sets. So all of the information is available to you to confirm that your microscope is actually compatible with the assay. And we recommend doing that before performing the assay.
Perfect. All right, next one here. So this person said that they do a lot of work with frozen tissue. Does the assay work with, will that work with frozen tissue?
So not at this time. We have not validated the assay to be compatible with frozen tissue. It is something that we look forward to doing in the future. At this time, all of the antibodies have been validated for use in FFPE, formalin-fixed paraffin-embedded tissue.
Excellent, great answer. What if the target that I'm interested in is not available in the panel builder?
Can you repeat the question? Sorry, I'm looking through some of the other questions.
Yeah. So what if the target I'm interested in is not available in the panel builder?
So if a target of interest is not available in the panel builder right now, we have a couple options. We do offer limited custom conjugation of our IHCP validated portfolio. Again, we don't want to allow scientists to utilize antibodies that have not already been validated for use in FFPE tissue because it may not work in SignalStar. So therefore, we enable custom conjugation of our IHCP validated portfolio, and you can look through our larger portfolio and see if there's targets of interest there. In
Right. Yeah,
addition,
that makes sense.
just to add, in addition, as we've demonstrated and showed data where we can combine H&E with after SignalStar on the same tissue, we've demonstrated that SignalStar is actually non-destructive to the tissue, unlike some other technologies. So there are opportunities to perform other assays potentially after SignalStar that would need to be further validated in your hands.
Fantastic. I think you just sort of covered this a little bit, but can I combine antibodies with other imaging assays?
So at this time, we don't have validation data to show you that we can do that. We know that the tissue is intact and that our assay is non-destructive and is not harsh on the tissue. So there's reason to believe that you would be able to perform direct immunofluorescence after SignalStar.
Excellent. So how long do I have to wait after staining to image these slides?
So one of the positive and benefits of SignalStar is that you don't need to run to the microscope because we ligate all of the oligonucleotides, the fluorescent oligos, after applying them to the antibody. We ligate them and stabilize that structure, and therefore we recommend anywhere within 8 hours after performing the assay to perform your imaging. But we know that it is stable for probably even longer than that.
Perfect. Yeah, nice to hear. Just going back to what you're saying about FFPE, how does the protocol account for autofluorescence in FFPE?
So because the SignalStar technology is an amplification-based technology, we're able to get above the autofluorescence signal. We're able to get, our signal is much brighter than autofluorescence due to the amplification. So we do not remove autofluorescence. We do not need to remove autofluorescence. We don't perform any kind of bleaching step beforehand. I think the assay would be compatible with that if that was something that scientists wanted to perform, to try to remove that autofluorescence using other mechanisms, but we do not need to do that in our hands with the targets in tissues that we've tested.
Excellent. All right. Do I need to optimize the antibodies to the specific tissue I'm using in my research?
We do recommend actually a little bit of optimization, just depending on the tissue and the quality of the tissue. You may consider doing a very tight titration of the antibody itself. In our hands and the tissues we're working with, we always use the recommended dilution, the 1:100. However, we do provide enough reagent in the kits and in your vials to be able to either use double the amount of antibody and obviously half the amount of antibody. And we recommend that if you are struggling with some background or with some weak signal, that you have the material to go ahead and give that a try and utilize a little bit of a range there. But often, and again, we've tested many, many tissues, we find the recommended dilution to be the most optimal.
Fantastic. All right, another good question here. What's an appropriate positive control to include in this assay? And are there multiple controls necessary?
So we do recommend that you identify your own positive controls for your tissues. So we recommend performing chromogenic staining, which is kind of, again, I've mentioned the gold standard for us, to confirm that the antibody is working in your tissue just for the detection of the antigen. So if your tissue, you would want to perform the chromogenic for each of your targets to confirm that your tissue actually expresses the target of interest, especially when you're first getting started. That would serve as a positive control, for example. And then you could move on to more experimental samples that may or may not have expression of each of those targets.
Excellent. All All right. It says from your slides, it looks like you cover not only human, but also mouse targets. It says, how is the overlap?
So some of the antibodies that I've put on that are on our list are cross-reactive in IHC, chromogenic IHC, to detect both human and mouse. We're currently performing the validation of these to confirm that in the SignalStar assay, we see the same thing, that we see that the capability to detect both, human in human tissues and mouse in mouse tissues. And so we're currently performing that validation, and the hope is by the end of the year, we'll be releasing a mouse reactive portfolio with up to 30 targets that will be available for detection in mouse tissues.
Great to hear. Very exciting. Are there any limitations for combining the antibodies for multiplex IHC, like CD markers and ILs?
We haven't seen any limitations in our portfolio. We've really combined these and tested these very thoroughly, combined them into many multiplex panels. For example, all of our validation happens in multiplex. So we're always performing an 8-plex. And as we do our titrations, as we test these targets across the different fluorophores, as we test them across the different imaging rounds, and as we test them in multiple protocols. So the amount of 8-plexes that they're put into really demonstrates that you can put these targets in really any configuration. And then again, the panel builder will help you put it into the configuration, and then you can always change it. We know that scientists may prefer a certain target in a certain channel, and it is available for you to do that as well.
Perfect. So I know you touched on this a bit in your presentation. Maybe you could elaborate. Do you have any recommendations for software for the data analysis?
Yes, absolutely. So we are currently using QUPath, which is an open source software I hope you may have heard of. And this is something that we're creating, currently in the process of creating a protocol for how to co-register these images and analyze these images using QUPath so that we can eventually provide that to the scientists performing SignalStar. It's not yet available, but I'm hoping to have it again before the end of the year. We've demonstrated it's also possible to co-register and analyze these images using Halo. And so that would be another one we would recommend. And we know we've also tried Visiopharm in-house, and they also can co-register and analyze these images very well. So pretty much any software that has the capability to co-register the two images and then perform the analysis as you would like should be compatible with this technology.
Awesome. Great to hear. Sarah, do you think this technology could be used for organoids research?
So we're still trying to understand that. That's something that's very interesting to us. And understanding how the organoids are prepared is really what we're going to be thinking about. So if the organoids are embedded in paraffin-embedded tissue or paraffin-embedded blocks, so treated similar to FFPE, then there's no reason to believe that this should work just as well in for staining organoid sections. If the staining is intended to be done on live organoids in culture, I think there needs to be a lot of validation still done to understand whether this technology would be applied to live cell imaging is basically what you would be doing at that time.
Fantastic. How did the oligos bind the antibodies?
So the oligos bind the antibodies due to the complementary nature of oligonucleotides. So basically we have our single-stranded oligonucleotide that is conjugated to the antibody using a proprietary chemistry. And then the oligos will come in, and they're just those complementary strands that naturally want to bind to their complementary, their opposite. And so that's the basis of the technology, is using the complementary nature of single-stranded DNA naturally wanting to become double-stranded DNA.
Fantastic. All right, another question here. How important do you see segmentation marker for your panels? Not sure what that means. Maybe you could parse it.
Absolutely. So I think what you're speaking to is using segmentation. Many scientists in multiplex imaging in general requires segmentation markers in order to get to that later analysis step, because you need markers in order to delineate this is the membrane, this is the nucleus. So our assay, we have the DAPI staining that obviously, on each image you would have DAPI in addition to your 4 targets. So that would help you start segmentation using a nuclear stain. We then personally in-house, we will utilize our membrane staining markers similar to, so if you're looking at, tumor, it would be pan-CK. And then if you're looking at T cells, you can use CD8, CD3. When you're looking, we have CD45, which is a marker of all immune cells. So you could utilize that to kind of delineate, this is the membrane of an immune cell. Pan-keratin, is on the membrane of a tumor cell, and that can help. You can train your algorithms to help perform segmentation. That's how we're doing it in-house currently.
Excellent. All right. Are there any issues with signal bleaching? And how soon do you think this might happen?
So fortunately, the Alexa Floor dyes are very robust, and so they aren't super sensitive to photo bleaching. We would imagine that over time this would happen. The longer you would do your imaging, if it goes over hours and hours, you might see a problem. But we've stored our slides even overnight at 4 degrees and kind of in the dark, and then image the next day. We don't have any problems with photo bleaching off of the microscope or just in our hands. And again, all of our staining, we image about 8 hours after we stain. And so just the cadence of how we do our imaging. So, and we have no problem with a robust signal after 8 hours after staining.
Perfect. And I think in the interest of time, we'll just have one last question. So Sarah, obviously this is a really new technology. And there's lots of exciting stuff on the horizon. Are there any developments coming up or avenues of research this is going to open up that you're particularly looking forward to seeing?
Yes, I'm very excited again about the mouse portfolio that we'll be releasing by the end of the year. So being able to really, enable translational researchers and doing this in higher throughput. We know that scientists that are doing mouse research, that it's not one piece of tissue that you can analyze and come to your conclusions. You often have, 40 or 100 or more tissues that you're working with. So being able to perform this technology without needing to optimize saves you that time to get to your higher throughput much faster and perform this assay over and over and over again on these, the amount of tissues that you're working, the mouse tissues that you're working with in translational research. So that's really exciting. And then on the far horizon, as many of the multiplex assays are moving towards, is we are planning to hopefully offer a neuro-oncology portfolio or neuroscientist science or neurodegeneration portfolio as well, so that we can help scientists working in neurodegeneration evaluate their tissues as well. And so that is something that we're excited about. In addition, something that makes cell signaling technology slightly unique or very unique is that we have a large portfolio of phospho antibodies, so PTM antibodies, post-translationally modified antibodies for detection of post-translationally modified proteins. And so we already have in our first cohort a phosphostat antibody for use in the SignalStar assay. And the plan is to expand upon that as well, because we have these antibodies that have been highly validated for use in chromogenic IHC for detection of phospho targets. And so we'll be adding that capability for the fluorescent readout, the SignalStar readout.
Perfect. All right. Well, Sarah, thanks so much for the great presentation today and all the insights in the Q&A. It's been really great talking to you.
Yeah, thank you so much. Thank everybody for your questions, and thank you for having me today.
Yeah, definitely. Big thanks to the audience for participating here. And in closing, we hope you enjoyed this Scientist.com and cell signaling technology webinar, and we'll see you again next time. Have a great day, everyone.